Thank you very much. Thanks for inviting me. My work hasn't really got a lot to do with oxidative stress. I look at the new anti-tumor drugs, and we've been looking at a number of natural products recently. So I'll be trying to bring us back to some of the discussions we've just had now with examples of what we're doing. Now, without going into too much detail, I think I can say that natural products, those that are used as anti-tumor substances, in reality have been found to have many mechanisms of action. And this is one of the reasons why, in some cases, they work. Research is increasingly looking at compounds that are multi-target, because attacking a tumor with just one target is very difficult due to the many different mechanisms of action that the tumor triggers. And the whole chapter concerning the microenvironment that was mentioned now is a very complex chapter concerning a wide range of things, whereas if the target is the tumor cell, that itself is very complex. Tumors come in hundreds with different molecular and biological characteristics, but they're a well-defined target, and the interaction between tumor cells, the stroma, the microenvironment, is only partly clarified. However, we do know that we're dealing, well, we know that we have to deal with angiogenesis, the formation of vessels, a number of different proteins, factors, some known and some not yet known, as well as different kinds of cells, fibroblasts, macrophages, that increasingly are important because, again, they produce substances, factors, they regulate survival and other mechanisms that may generate proliferation, progression, metastases, and so on. Therefore, when we study a new compound, we look at the mechanism of action against the tumor cell, but we should also take into account many other things that are going on. And just to give you an example of substances that we wouldn't have suspected having other functions when they went into clinical trials that have nothing to do with the direct action. The example is camptothecins, which at the transcription level are H1 alpha. This is a very important factor for angiogenesis for many tumor biology aspects, and as I said, this is very important. Taxanes as well have an ability to inhibit angiogenesis, and this was described for many years by many laboratories. Volkmann, who was the forefather of angiogenesis, and now there is clinical evidence for this effect. Data on ovarian cancer shows that patients who are resistant to taxanes administered with the typical schedule may instead be sensitive when the taxane is administered with a different schedule and with a stronger angiogenic effect. And there are many clinical trials underway on that subject. So, as Dr. Morazzoni was saying before, Endena has worked on taxanes and obtained and characterized a number of new taxanes that are currently being investigated. And these are interesting studies because it may well be that within a certain category or class of drugs, there might be certain structures that are better able or less able to kill cells and a greater ability to have an anti-angiogenic effect and to modulate the microenvironment. For example, this is a substance that's been studied in our lab in conjunction with Endena, and it has effects on the endothelium versus direct, very much greater direct cytotoxic effects. However, I'll now go on to substances that we have studied in our lab that may be taken as prototypes for substances that may have cytotoxic effects as well as effects on the host and other mechanisms. These are natural products. This is a natural product And in vivo, it's shown a number of interesting effects. And this has led to the clinical and preclinical, preclinical and clinical development of the drug. The substances, we weren't familiar with the cytotoxic effect, but we did see the considerable oxidative stress and a number of genes associated with the effect. It is particularly powerful on certain types of cells, leukemia and lymphoma in particular, primarily due to a particular loop that is blocked and that I'll be talking about in a minute. For your information, this is the drug that went into clinical trial, into clinical use. It was effective on myalgia and also showed some effects on certain cancers. I won't go into too much detail, but on certain tumors, there were partial responses. This is a study on myeloma, for example. Now, the substance, aplidin, is really stable when it is administered, and our group saw that one reason why certain types of cells are affected by it is because it blocks the release of VEGF in areas where there are this loop. You can see the mechanism being blocked, leading to regulation of the receptor and then induction of apoptosis and growth arrest. Various cell lines have been studied, and it's interesting to see, I don't want to go into too much detail, but that in fresh cells,